Hi, good evening. My name is Doris Valencia Pico. I am here to talk about hemoglobin. My presentation is called A Protein of Life, just because it really helps sustain life in many ways. So, objectives for this presentation: you will learn the basic hemoglobin function, structure, and properties. You will learn how oxygen binds to heme. You will learn the structural changes that occur during gas exchange. You will learn the binding capacity of oxygen to heme in the lungs and how that differs from that in the capillaries. Also, it is my objective with this presentation that you will learn how carbon dioxide interacts with water and how that affects the capillaries. So what is hemoglobin? Hemoglobin is an allosteric protein. It is also a quaternary structure composed of 4 polypeptide chains, 2 alpha and 2 beta. Each of them is bound to heme. It's also dimers of alpha 1 and beta 1 and alpha 2 and beta 2. Now we know that fetal hemoglobin is alpha and gamma dimers. So here we have the structure of hemoglobin. We have the beta 1 with the alpha 1 and the alpha 2 with the beta 2. So how does oxygen bind to heme? Heme binds oxygen to a ferrous ion on its center. Thus, a molecule of hemoglobin can bind up to 4 molecules of oxygen. This is called a cooperative process. We know that about 98% of saturated hemoglobin with oxygen is in the lungs, and the oxygen saturation drops to 32% in the tissues. So basically that 66% of oxygen they get released in the tissues. This behavior begins in the lungs, where hemoglobin becomes approximately 98% saturated with oxygen. We see a graph of cooperativity, and basically this is a very important process during the oxygen binding to the hemoglobin. Let me go back. Binding at one site changes the conformation of the protein molecule. That makes it easier for more oxygen molecules to bind in other sites. This formation of the hemoglobin oxygen complex is called oxyhemoglobin, which is equal to our state. Here I wanted to just share a fun fact. Allosteric regulators released at the tissues can improve the oxygen release. So we have the cooperative process, but we also have the 2,3 biphosphogylacerate, which is very important in helping the hemoglobin reduce its affinity for oxygen. This reducing affinity for oxygen allows hemoglobin to release that 66% of oxygen into the tissues that we talked about earlier. The structural changes that occur, as we were talking about earlier, is that binding at one site changes the conformation of the protein. So we know the formation of the hemoglobin oxygen complex is called oxyhemoglobin, and this is an R state. Now the deoxyhemoglobin is a T state. Basically, this is less chemically active. So what happens when the oxygen binds to hemoglobin? So basically, iron ion moves into the plane of the porphyrin. The histidine residue is bound to the fifth coordination site and moves with it. It forms kind of like an alpha helix. Then the carboxyl terminal of the alpha helix lies in the interface between the two alpha-beta dimers. This therefore causes substantial changes in the structure, leading T to R transition. Then the alpha to beta to rotate 15 degrees, allowing rearrangement of the dimers to communicate. Therefore, one oxygen on one subunit changes from T to R. And then the 2,3 BPG binds to oxyhemoglobin. So 2,3 BPG is not the only allosteric regulator. So here I have a graph, a picture, about what happens in this structural change. So here we have the beta and the alpha, and how they become, from the oxy T, it rotates 15 degrees to become the oxy R. In that way, by changing the position, this bonds between hemoglobin and 2 and 3 must be broken. So the 2,3 BPG must be expelled. Here we have another graph of how the movement of the iron ion on the oxygenation binds to the iron associated histidine residue in the porphyrin ring. So we see the oxygenated heme versus the deoxygenated heme. This is irreversible depending on oxygen partial pressure in the tissues. Oxygen pressure is 100 torr. The associated oxygen in the tissues is basically 20 torr. So we can see the difference in partial pressure of oxygen in the lungs versus the tissues. So oxygen binds in the lungs where, again, the pressure is 100 torr, and this associates in the tissues where the partial pressure of oxygen is 20. Hemoglobin is responsible for most of the oxygen carrying function in the body. However, it also carries carbon dioxide. And how does that happen? How carbon dioxide plus water influence in the oxygenation of the body, and how hemoglobin carries all this, the oxygen and the carbon dioxide. So what happens is the carbon dioxide binds to the polypeptide part of the hemoglobin and not the heme, but its binding changes the conformation of them, and therefore it decreases its affinity for oxygen. In other words, the two gases compete for binding on hemoglobin. Oxygen binding is optimal in the lungs, and carbon dioxide binding happens in the tissues. Carbon dioxide is then transported in the blood in the form of bicarbonate ions. The conversion of carbon dioxide into carbonic acid dissociates into bicarbonate and hydrogen ions, which it is catalyzed by an enzyme called carbonic anhydrase. Then the bicarbonate ions diffuse into the plasma to be exchanged with chlorine ions and hydrogen ions. This bind to hemoglobins that have released oxygen. Red blood cells reach the lungs. This reversal happens when there's high oxygen pressure. So that high oxygen pressure favors its binding. Then the carbon anhydrase converts bicarbonate and hydrogen ions back into carbon dioxide, which we breathe out. We talked about how the polypeptide binds to, the carbon dioxide binds to part of the polypeptide, etc. So here I made this diagram. So what happens, like the sequence of the process. So oxygen binds in the lungs, carbon dioxide in the tissues. Carbon dioxide is transported in the blood in the form of bicarbonate ions. Then it converts from carbon dioxide into carbonic acid, which dissociate into hydrogen ions and bicarbonate. They get catalyzed by the carbonic anhydrase, and therefore here we have a picture of what happens at the tissues. So the tissues, we have a high concentration of carbon dioxide. This carbon dioxide is released into the red blood cell, where it converts the carbon dioxide plus water, then the bicarb into free hydrogen ions. These hydrogen ions cause that pH to change into acidic or low pH. So the Bohr effect basically shows the hemoglobin response to carbon dioxide with a decrease in oxygen affinity. So when there's an increase in carbon dioxide molecules in the tissues, this facilitates more release of oxygen. And that is because carbon dioxide decreases the pH. Therefore, there's a shift to the right on the hemoglobin dissociation curve. So here we see as the partial pressure of oxygen decreases in the tissues, there's more buildup of carbon dioxide. Therefore, the pH decreases and there's a shift to the right. However, when the partial pressure of oxygen increases, there's a shift to the left. Basically, the hemoglobin doesn't need to release so much oxygen versus when the carbon dioxide is high in the tissues. Of course, here are my references, and we now understand why I call my presentation hemoglobin a protein of life, because it really regulates not only provides oxygen to our tissues, but also provides a balance in pH as well. Thank you so much for your time. Bye bye.